It's difficult to modulate gene expression in human islets. Also, gradual decline of the function in human islets pose major challenges after modulation of gene expression. By combining the lentivirus mediated shRNA transduction and the formation of human pseudoislets, this protocol allows efficient downregulation of target gene while preserving islet function during prolonged culture.
To begin, gently swirl the shipping bottle to keep islets in suspension. Transfer the shipping medium containing islets to a 50 ml conical centrifuge tube. Let the tube sit in a biological safety cabinet for 15 minutes so that islets settle to the bottom of the tube. Remove the shipping medium gently using a capillary pipette without disturbing the islet pellet. Resuspend the islet pellet in CMRL medium with 1% human serum albumin to a concentration of 400 islet equivalent per milliliter. Transfer islets into a non-tissue culture treated dish and culture them at 37 degrees Celsius in a 5% carbon dioxide incubator overnight. After overnight culture, transfer human islets into a 15 ml conical centrifuge tube. Centrifuge at 250 times G for 5 minutes in a swinging bucket rotor, and aspirate the medium with a capillary pipette without disturbing the islet pellet. Wash the pellet by adding 10 milliliters of PBS to the tube.